Hello, my name is Dr. Roderick L. Roll, and today I will be talking about molecules and compounds. In order to make molecules and compounds, we will have to use different atoms. In this slide, we have 3 atoms: hydrogen, helium, and lithium. If we joined lithium and helium and hydrogen together, we could make a molecule. Molecules are composed of 2 similar atoms, or they can be composed of 2 different atoms. These groups of atoms will be held together by chemical bonds. In the image below, I have several examples of molecules of an element. The first one is O2. O2 would be 2 oxygen molecules covalently linked together. Lower in the image, you'll see 8 sulfurs covalently linked together. These are same or similar atoms, so therefore when they are linked together, they will be molecules of an element. The next one is molecules of a compound. This compound will be composed of different elements. An example is water and methane. Water is composed of 1 oxygen covalently linked to 2 hydrogen. This will represent a molecule of a compound. Now we will talk about mixtures. Mixtures are 2 or more elemental components physically mixed together. They are not chemically bonded. So you will not see an ionic bond or a covalent bond to hold these mixtures together. They are just side by side as atoms and elements. So mixtures are not chemically linked. So we will talk about 3 types of mixtures: a solution, a colloid, and suspensions. These mixtures will either be homogeneous or heterogeneous. Homo means the same and hetero means different. So a homogeneous mixture has similar parts, or they are of consistency, uniform consistency. The example I love to use is one of my favorites: Crown and Coke. This would be a homogeneous mixture. The parts are of uniform consistency. The Coca-Cola and the Crown looks consistent. The second example is of a heterogeneous mixture. Water and oil would be a heterogeneous mixture. The first mixture that we will talk about is solution. A solution is a homogeneous mixture. It has 2 components: a solvent, which is typically water, and a solute. The solvent is the thing that is present in greatest amount, whereas the solute is the thing that is present in the least amount. So an example is sweet tea. The water in the tea will be the solvent. The tea or the sugar would be the solute. Now, how do we calculate percent? So when we're looking at these solutions, you always want to understand how to figure out the number of particles that are in these solutions. So for example, if we want to know how much sugar is in the sweet tea, we can use a unit called percent. We can also use mole, but let's just talk about percent now. So a percent is based on parts out of 100. So in this image, I have an example of how to calculate a 10% solution of glucose, a 20% solution, and a 85% solution. In a 10% solution, you would just measure 10 grams of glucose and put it in a graduated cylinder. Then you would add the remaining parts with water. You will bring the volume up to 100 to equal 100 parts. So therefore, you would have added 90 milliliters of water. Be able to calculate 20% and 85%. How did we know that 1 milliliter is equivalent to 1 gram? Previously in lab, you put a graduated cylinder on an electronic balance and you weighed the volume of water and you saw that it was a 1-to-1 ratio. So therefore 1 milliliter of water is equivalent to 1 gram. So again, you put a weigh boat on an electronic balance, you measure out 10 grams of glucose. You put it in a flask, a graduated cylinder, or a beaker. Then you would bring the volume up to 100 with water. Therefore, you would have added 90 milliliters of water. The second mixture that we will talk about is a colloid. A colloid is a heterogeneous mixture with solutes that are invisible. This means when you make jello shots, you would take powdered jello and mix it with ethanol. Let's just make regular jello. You would take jello powder and mix it with water. The jello is invisible. The jello granules will be invisible and they will not settle to the bottom of the container. This means it is a colloid. Another example of a colloid is the cytosol. This is the substance that is between the cell membrane and the nuclear membrane. All of that is a gelatinous material that is jello-like in appearance. The third example of a mixture is suspension. A suspension is a heterogeneous mixture with visible solutes that tend to settle out. The example we will use is blood. If you were to drain some of the blood from your femoral artery and put it in a test tube and let it sit, it would separate. The heavier red blood cells are called erythrocytes. They would settle to the bottom, and the water part of blood would be on the top. That would contain the plasma. So therefore, you would have distinct separation of parts, visible parts. This is the example of suspension. Now we're going to compare mixtures versus compounds. So to conclude, with mixtures, no chemical bonding takes place. Most mixtures can be separated by physical means. Let's look at a few of those physical means before we move to look at examples of compounds. A mixture of iron and sulfur can be separated by using a magnet. So on the periodic chart, you will notice that some elements are metals and some are nonmetals. So if we had a mixture of the two types, we could easily separate the metals from the nonmetals just by using a magnet. So this is magnetism. This is one way to separate a mixture. A second way to physically separate a mixture is by distillation. This uses heat to separate the mixture. This looks like one of the distillers that we used when I was working on my PhD. We used a distiller to make distilled water. Distilled water has been separated from its other physical parts, like heavy metals and other elements that could be in water. So when performing an experiment and you need ultra pure water, you would distill it. This distiller is going to heat up water to such a temperature that it evaporates. The evaporated water will be collected, and the leftover would be the impure particles, like elements, like lead, like iron. So distillation separates parts. Here is another example of distillation separating ethanol from water. In the bottom image, you notice that ethanol has a lower boiling point than water. So therefore, the ethanol will boil and be released from the solution, from the mixture, and it will be collected in this condenser. Then you could physically separate ethanol from water. The third physical way of separating mixtures is by filtration. Filtration utilizes exclusion. Exclusion is going to leave out the larger particles that cannot squeeze through the filter. So in the top right image, that is a filter paper that is embedded into this plastic mesh. When you pour the solution into a funnel that contains that filter, everything that is larger than the pores in the paper will be trapped above the paper. This is used in air conditioning. This is used in hospitals when you hear about a HEPA filter. That is how you purify a room's air source, is through taking the air through a filter that contains this paper that will trap the larger particles as it tries to come through. The fourth method of separating mixtures physically is crystallization. Crystallization uses attraction to separate. So in this example, I used to make rock candy. That was my first science experiment. How did I do it? I made a viscous solution of sugar water. Then you stick a stick into the sugar water and allow it to sit. The sugar is attracted to the stick, and then other sugar molecules are attracted to the sugar that's stuck to the stick. So eventually, through aggregation, you create rock candy. This is crystallization using attraction. Chromatography utilizes attraction also. So this is our fifth method to separate mixtures. So paper chromatography is something that we use in science a lot. You can take a mortar and pestle and grind up spinach leaf or some other kind of plant leaf. You will get a extract. Then you could take the extract and dab a little of the extract onto the paper chromatography. Then you take the paper and stick it into a test tube that has acetone at the bottom. The acetone will migrate up the paper. It will therefore separate out the different pigments. It will separate out 4 pigments: chlorophyll B, chlorophyll A, xanthophyll, and keratin. If you notice in this image to the right, chlorophyll B has the strongest affinity to the paper, whereas keratin has the least affinity to the paper. So this helps to separate out the different components that are in that chlorophyll or green extract that you achieved from squishing, grinding, pulverizing your Spanish leaf. So to conclude, mixtures can be heterogeneous or homogeneous. We will now talk about compounds. Compounds cannot be separated by physical means. All compounds are homogeneous mixtures, and they are chemically linked. This means the electrons in the outer shell of these elements are being shared or being stripped away. An example of a compound is steel. Steel is composed of iron and carbon, 2 elements from the periodic chart.